Hello everyone, welcome to our webinar. It's about chemical mediators. It's presented by me, Mohanad Riyadh, with my colleagues Abdalrazaq, Khalid, and Dhuha Sufyan. It's supervised by Dr. Zahraa, Marwan. I'm going to start with the definition, or what's the chemical mediator? It's a chemical substance that mediates a specific response in body tissues. So it doesn't make the response itself, it mediates it. It's something like messaging system between cells, and generally chemical mediators have a short lifespan. In this video, we are going to know more about the roles of chemical mediators. When foreign invaders such as bacteria or virus get into our bodies, they can cause infections. These events activate our body defenses, such as white blood cells. A certain type of white PCs, for example, helper T cells, release chemical mediators that can attract more inflammatory cells to the site of infection. These additional white blood cells or inflammatory cells attack the invading bacteria or virus. Some mediators can make cells go into apoptosis through their death receptors. Another mediators can activate other white blood cells, cause them to multiply, and even stimulate them to create markers called antibodies. Antibodies attach to the bacteria or virus, marking them as target for our immune system to destroy them. Those mediators can travel throughout the body, therefore cause systemic manifestation. Chemical mediators could be classified into two main groups according to their source of origin. They are either cell-derived or plasma-derived. Cell-derived mediators are subclassified into two groups: preformed mediators or newly synthesized mediators. Preformed, they are already made and stored in secretory granules. Newly synthesized, they are synthesized de novo, made if needed. Preformed, such as histamine and serotonin. Newly synthesized, such as cytokines, for example interleukins, eicosanoids, prostaglandins, nitric oxide, reactive oxygen species, leukotrienes, and the platelets activating factor. Plasma-derived, such as complement system activation and factor 12, Hickman factor activation. Plasma-derived chemical mediators are secreted by liver into plasma as an inactive precursors. We are going to discuss topics appear in red, starting by histamine. Histamine is produced from the essential amino acid histidine by mast cells and basophils found in connective tissues nearby inflammation. Here is the reaction of decarboxylation of histidine, which produces histamine, and a microscopic photo for our pretty mast cells. The main function of histamine is increasing the permeability of the capillaries. Here is a blood vessel under the effect of histamine. Endothelial cells contract, and spaces or gaps between them are opened. This leads to increased capillary permeability that allows fluids and proteins to pass from the blood into the tissues. And in process called diapedesis, neutrophils squeeze through the endothelial gaps to reach the site of infection. Increased vascular permeability causes fluids to escape, as we said, which leads to the classic symptoms of allergic reaction, runny nose and watery eyes. The histamine is also the mediator of itching. But how can histamine cause itching? Here is a simplified diagram of the nerves of the skin. A group of them respond to histamine. They are called histamine-triggered neurons. Each nerve sends signals to the spinal cord. The itch signal is highly processed in the spinal cord and goes to the brain, which makes us to scratch. Scratching, or in another word, pain, blocks the itch normally. But unfortunately, scratch leads to compromised skin barrier or damage to the skin, which leads to further inflammation and further itching, and this is known as itch-scratch cycle. And now we shift to cytokines. Cytokines are a broad and loose category of proteins. The first identified cytokine was interferon alpha in 1957. Cytokines are divided into subsets or families. Interleukins, human necrotic factor, chemokines, and interferons are all examples of cytokines. Here is an animated video of a macrophage releasing cytokines. This is known as cytokine storm, which is the main cause behind the severe symptoms of inflammatory diseases such as COVID-19 nowadays. And here comes the first example of cytokines. Interleukins, and as the name indicates, inter, between, and leukin, leukocytes. They play a role in intracellular communication among leukocytes. Up to 38 interleukins have been so far identified, and they are numbered according to the order of discovery. Interleukin 1 is our first example of interleukins. Interleukin 1 is produced mainly by macrophages, as well as monocytes, lymphocytes, neutrophils, and keratinocytes. It binds to the interleukin 1 receptor 2, stimulates fibroblast proliferation, increases blood neutrophils, activates lymphocyte proliferation, and induces fever. Now we are going to discuss how interleukin 1 induces fever. As we all know, the thermostat of our body is the hypothalamus. Interleukin 1, interleukin 6, and tumor necrotic factor, which are known as pro-inflammatory cytokines, affect hypothalamic control of body temperature, which leads to increased set point value and then fever. But there is a problem. The blood-brain barrier only allows molecules around 500 Dalton to pass through. How can the interleukin with 17 kilodalton to reach the hypothalamus? Actually, interleukin 1 affects a specific area in the brain called vascular organ of lamina terminalis, or VOL, and this is an area of the brain with no blood-brain barrier. Our second example of interleukins, interleukin 6 is an important mediator of acute phase response. The sources of interleukin 6 are macrophages, T cells, B cells, smooth muscle cells, and others. Interleukin 6 is an important mediator of fever. It's changing the body temperature set point in the hypothalamus as in interleukin 1. And in addition of that, in muscles and fatty tissues, interleukin 6 stimulates energy mobilization that leads to increased body temperature. Interleukin 6 also stimulates osteoclast formation, and smooth muscle cells in the tunica media of many blood vessels also produce interleukin 6 as a pro-inflammatory cytokine. Here is a table for other interleukins. Interleukin 3 from T cells, macrophages, and natural killer cells. It is a hematopoietic growth factor. Interleukin 14 from T cells. It plays a role in proliferation of activated B cells. And interleukin 24 from melanocytes, T cells, monocytes, and normal human keratinocytes. It has a role in tumor suppression. And now my colleague Abdalrazaq is going to continue.
Hello everyone, I am Abdalrazaq Khalid, and I will take you on a journey with an important molecule inside our body that's directly affecting its function or indirectly by its pathway. This molecule is arachidonic acid. Arachidonic acid is a non-essential fatty acid composed of 20 carbon atoms and is fixed within the cell membrane lipid bilayer as a fatty acid chain. So, it has many important functions, including cell membrane integrity, ion channels function, and nerve impulse. My concern today is its function as a chemical mediator and as a parent molecule for a family of mediators called eicosanoids that has a major role in inflammation, thus they are called local hormone. Eicosanoids, their name defines their structure because eikosa is a Greek word for 20, the same number of carbon atoms in their structure, and noid is a suffix used for fatty acid. Here we have a diagram that shows arachidonic acid pathway, which can follow two routes, either being converted into prostaglandins or leukotrienes. We will take this pathway in details in just a moment. But before that, we may ask, what causes the arachidonic acid to be released in the first place in an amount that can cause inflammation? We can say that whenever there is a damage to the cells because, for example, an infectious agent, trauma, radiation, or even autoimmune disease, the damaged plasma membrane will expose the arachidonic acid to an enzyme called phospholipase A2 that will set free arachidonic acid to be diffused away and induces formation of many chemical mediators that will affect the nearby vessels and nearby immune cells to start the inflammation. So, this explains the fact that whenever there is a damage to the cell, there is a release of chemical mediators that will induce inflammation. Now back to our friend arachidonic acid. As we can see here, the arachidonic acid is fixed within the cell membrane lipid bilayer and can become free by the action of enzyme phospholipase A2. Arachidonic acid now, it has two routes. Depending on the enzyme, it can be converted into prostaglandin G2 by the action of enzyme cyclooxygenase or to leukotrienes by 5-lipoxygenase. We can see the complex molecular structure of these three enzymes. Starting with the prostaglandins. Prostaglandin G2 is an unstable molecule, so it will be converted into prostaglandin H2 that will give rise to prostaglandin E2. Prostaglandin E2 will promote pain, fever, vasodilation, and all of these are the features of inflammation. It also promotes mucus production, especially in our stomach. Prostaglandin H2, it also can give rise to two different mediators, depending on the site. So, if it was in the platelet, it will give rise to thromboxane A2. And as the name implies, thrombo, it means a thrombus. So, it will induce thrombus formation by inducing vasoconstriction and platelet aggregation. On the other hand, prostaglandin H2, it can be converted in the endothelium to prostacyclin. Again, as the name implies, prostacyclin. That means we need the blood to cycle inside our blood vessels. So, it promotes what? You guessed it, vasodilation and prevent platelet aggregation. You may notice that we have the same parent molecule that give rise to two mediators that are completely opposite in action. So why is that? Actually, because it is necessary for maintaining balance and achieving the body needs. For example, if we had a bleeding and we need to stop it. So the body shifts the pathway from prostaglandin G2 into prostaglandin H2, then thromboxane A2. So more thromboxane A2 will be produced and less prostacyclin will be produced in that area of bleeding. As a result of that, we will have vasoconstriction and platelet aggregation that will induce the formation of thrombus that will stop the bleeding. Now we continue our pathway, move to the other half where the arachidonic acid being converted into leukotrienes by 5-lipoxygenase. Leukotriene give rise to B4 that promotes chemotaxis and C4D4E4 that promotes bronchoconstriction. Leukotrienes can also give rise to a family of mediators called lipoxenes, which their main function is inhibition or resolving inflammation. This is also to maintain the balance and to prevent the excessive inflammatory response. At this point, I have to say, congratulations, we have finished the arachidonic acid pathway. But you may think, why I explain this particular pathway in details? Actually, there is a great importance of this pathway in our medical practice. For example, we know when treating an acute inflammation such as anaphylactic or septic shock or chronic inflammation such as rheumatic arthritis and many autoimmune diseases, we use steroids such as corticosteroid or hydrocortisone. On this pathway, we can explain why. Because steroids will inhibit the action of phospholipase A2, thus inhibiting the formation of arachidonic acid, therefore inhibiting the entire pathway, thus reducing and inhibiting the inflammation. We have another group of drugs called non-steroidal anti-inflammatory drugs that inhibit the inflammation by inhibiting the cyclooxygenase enzyme and blocking this pathway and preventing the formation of all of these mediators. So this results in inhibition and reducing the inflammation. We have also aspirin, which is one of the non-steroidal anti-inflammatory drug family, is also used as a prophylaxis against thrombus formation, because when blocking this pathway, thromboxane A2 will not be produced, thus decreasing the chance of clot formation. So aspirin can be used as a prophylaxis for MI and stroke. However, there is some disadvantage of using these drugs because when blocking this pathway, prostaglandin E2 will not be formed, so mucus production is inhibited, thus may lead to a peptic ulcer. On the other hand, non-steroidal anti-inflammatory drugs and aspirin is contraindicated in asthmatic patients. But why? Because when we are blocking this pathway, all the arachidonic acid will shift into formation of leukotrienes, part of which is C4, D4, E4 that promotes bronchoconstriction, and this is very bad for asthmatic patient. Now, continue as a summary. The functions of eicosanoids. Prostaglandin E2 promotes pain, fever, mucus production and vasodilation and uterine contraction, and even increasing the sperm motility. And just an extra information, prostaglandins, it sounds just like it's related to the prostate gland. Yeah, it's true, because first the prostaglandin were isolated from a specimen of a prostate gland secretion. So they name it as prostaglandins. After then, scientists found that the prostaglandin is distributed all over the body tissues. And you may notice that we have E2 leukotriene, E4 leukotriene, D4 leukotriene, B4. What does these numbers mean? Actually, it's referring to the number of double bond in between carbon atoms in their structure. Now we continue. Prostacyclin, as we have taken, it promotes vasodilation and prevents platelet aggregation. I have a microscopic video that shows the vasodilation process. As we can see here, this is real blood vessels is vasodilating. This speed is 8x. We also have thromboxane A2 that promotes vasoconstriction and promotes platelet aggregation. We have leukotriene B4 that promote chemotaxis and leukotriene C4, D4, E4 that promote bronchoconstriction. I have another microscopic video for chemotaxis. As we can see here, neutrophil is chasing Staphylococcus aureus until it's being catched and in the cytosol. Now my colleague will continue.
Hello everyone, I am Dhuha Sufyan and I am talking about chemical mediators and the resolution of inflammation. Firstly, what's acute inflammation? As we all know that acute inflammation is a protective mechanism, experienced as heat, redness, edema, pain, and loss of function, in which the body defenses against invading microbes, noxious stimuli, and tissue injury. It assists in conserving our health and maintaining homeostasis. But unfortunately, excessive irregular inflammation contributes to many widely occurring chronic diseases with their complications, such as osteoarthritis with its pain, COPD with its annoying bad effect on the respiratory mechanism, and even cancer can result from that continuous irregulated inflammation, so improvement is needed. But how does this improvement occur? Or how does inflammation end? Actually, it ends by a process known as resolution. The term resolving and concept of promoting the disappearance of inflammation traced to the 10th century in the Qanun of Medicine, which is written by the great scientist Ibn Sina. And nowadays, pathologists describe resolution as a complex, highly active and organized process that happens slowly, aiming to end the inflammation naturally without compromising the immune system and without risky side effects. It involves decomposition and breaking down the products of inflammation, ceases inflammation, and returns the body back to the normal. And as the inflammation begins with mediators, there is also special mediators for resolving it, named as SPMs, or specialized pre-resolving mediators. They are newly discovered, naturally occurring mediators considered as part of the immune system, functions to actively keep the inflammatory response within its physiological boundaries. These mediators are lipoxins, resolvins, protectins, and mycins, which are started to be produced during the transition from inflammation to resolution, when switching between the mediators takes place, in which the initial formation of pro-inflammatory mediators induce the production of enzymes that enable the reprogramming of leukocytes to generate SPMs from the same polyunsaturated fatty acids. SPMs don't inhibit inflammation, but rather help facilitate or support the clearance of inflammatory components and thus resolve naturally the inflammatory process. SPMs limit the further recruitment of PMNs but stimulate the recruitment of macrophages that organize the clearance of the inflammatory exudates containing microbes and dying neutrophils. SPMs stimulate macrophage phagocytosis, resulting in the removal of apoptotic cells and/or microbes and the clearance of PMN. A key component of the resolution process is enhancement of macrophage apherocytosis and bacterial killing, and to this end phagocytosis has been established as a biological indicator of resolution in experimental, clinical, and animal studies. SPMs reduce pain and also promote tissue regeneration. SPMs function to actively keep the inflammatory response within physiological boundaries and accelerate the return to homeostasis. SPMs don't inhibit inflammation, but rather support the unique and distinct resolution phase of the inflammatory process. So, there are many benefits of SPMs, including reduce pain by treat its causes, promote tissue repair and regeneration after trauma or surgery, lowering the requiring for antibiotics, and protect you naturally from getting into chronic inflammation and cancer. Thank you for reaching with us this far. Hope you enjoyed, and we wish you the best.